Welcome back. In this video, we're going to talk about a very strange aspect of amino acid catabolism. And it's strange because up until this point, we've only dealt with catabolic pathways for L amino acids. But keep in mind that in some bacterial structures, we have D amino acids. Well, what happens when we have a D amino acid? Well, it turns out that the enzymes we have for L amino acids can't really deal with them very well. So we have to use other means to catabolize D amino acids. So what I want you to bear in mind is I haven't drawn or indicated stereochemistry here, but assume that this amino acid right here is the D isomer. Okay, just make that assumption. Okay, and so what we have to understand about the D amino acid oxidase is it requires molecular oxygen, a critical serine residue, water, a D-amino acid, and FAD. So this flavin that's up here at the top, this is flavin adenine dinucleotide. Okay, and obviously the R prime up there is the ribotyl plus the ADP down there. Okay. So the very first step in the mechanism is going to be a deprotonation of a critical serine residue in the active site, okay? And it's going to be water that's in the active site that does this. And this water is in there pretty tightly. It's not like a water that just automatically comes in. It's in the active site for good, okay? So that water comes and deprotonates the serine residue. And as the serine residue gets deprotonated, it abstracts a proton from the alpha amine of the amino acid. And that causes the formation of a Schiff base by forming this double bond between the nitrogen and the alpha carbon. Keep in mind that this is the alpha carbon right here, right? And that forces the alpha hydrogen off as a hydride. And it's going to come and attack this nitrogen on the FAD ring. And what ends up happening is you get a double bond rearrangement. So this pi bond ends up right here, and this pi bond ends up as a lone pair on this nitrogen. And so what you should expect to see in the next FAD derivative structure is a negative charge right here on that nitrogen, and we do see that. Okay. Now, in this step of the mechanism, what's about to happen is we're about to see our first instance of flavin electron transfers using radical electrons. Okay, so what's effectively going to happen is this double bond that's right here, this double bond right here, this double bond is actually going to deliver one of its electrons onto molecular oxygen. Now, what I want you to bear in mind is this. Keep in mind, and they may not have taught you this in biochemistry or gen chem or whatever, but this is actually the correct structure of molecular oxygen. The reason they draw it with a double bond ordinarily is because when you do the calculations, it has a bond order of 2, but that is not the correct structure. In fact, what you can really view it as is if you had the double bond oxygen, what effectively happens is you have a homolytic bond cleavage of the pi bond. And so each one of the electrons from that pi bond ends up on the opposite oxygen atom. So this right here is actually the correct structure. And this is going to be the structure by which the electron is abstracted from the FAD. Okay, so these electrons kick back up onto as a pi bond on FAD. And what's going to happen is, and let me do this in blue, this pi bond right here, one of the electrons is going to end up as a radical right there. And specifically, it's on this carbon that I just highlighted. The other electron that's part of the pi electron system is going to end up adding onto molecular oxygen right here. So your initial product, as we'll see in the next picture, is actually going to be superoxide. Okay, so this molecule right here, this is superoxide. Okay, now at physiological pH, and especially in the active site here, superoxide will not pick up a proton even though it has a negative charge. And the reason that in general it doesn't pick up the proton is because one of those electrons can sort of be in a pseudo resonance between both oxygen atoms. So it's not basic enough to pick up a proton. But what's going to happen essentially is the superoxide, at least the oxygen that has the radical electron, is going to couple with the radical electron that's on the FAD. Okay, so this electron right here is going to couple with this one right here. And so what you should expect to see now is a peroxide linkage now attached to the FAD. Okay, and as that linkage is being formed, as you can see right here, the electron lone pair that's right here on the other oxygen atom is going to re-abstract a proton from the hydronium that we generated in the first step. And of course, that regenerates the water molecule that we had at the beginning. Okay, and keep in mind that water molecule is in there pretty tightly. What we're about to see is called a cyclic intramolecular elimination. So these electrons right here are going to come out and abstract the proton from this nitrogen, and that's going to force these electrons between the hydrogen and the nitrogen to form the double bond right here. And as a result, you end up losing hydrogen peroxide. Okay, you end up losing hydrogen peroxide. So we observed that product right here. This is hydrogen peroxide. And in the process, what that does is it regenerates our FAD. And that's this right here. This molecule right here is our FAD. Well, now we've regenerated FAD, but there should be a burning question in your mind. And that question is, what happened to the original D amino acid? Remember that we formed the Schiff base in the first step of the mechanism. Well, we still have that Schiff base, and it exits the active site. And what I want to be perfectly clear about is the step that we're about to see is going to be a Schiff base hydrolysis, as you might expect, but this is not catalyzed by the enzyme. This actually is proven to occur once this molecule, this protonated Schiff base, once it leaves the active site. So once it leaves the active site, it's going to undergo a spontaneous Schiff base hydrolysis. Okay, let's look at that now. Okay, now if you need help predicting products of Schiff base hydrolyses, I highly recommend that you do that. At this point, I think it's something you should be able to do off the top of your head. So effectively what's going to happen is the water is specifically going to attack this carbon right here. It's going to attack this carbon, and when you do the complete Schiff base hydrolysis, you should get an alpha keto acid. So this molecule right here, this is an alpha keto acid, and you should get ammonia. So these are your two products of the Schiff base hydrolysis after the D-amino acid oxidase mechanism. Okay, in a separate video, we'll actually look at the catabolism of those alpha keto acids, and to some extent, you already know them. Well, now we've seen the mechanism of D-amino acid oxidase, and the question is, why would you ever want to learn about this? Well, let's think about something. Let's say I want to go eat an apple, and actually it really doesn't matter if you cook the food or you eat it cold or whatever. It really doesn't matter. On some level, always, you are going to be ingesting bacteria. Okay, you can't avoid it. Even if you cook something, there's an astronomically slim chance that you're not getting bacteria. Okay, you are constantly going to be ingesting bacteria. In fact, you might be breathing in bacteria as we speak, as terrifying as that sounds. Okay, and whenever you get the bacterial cells in your stomach, obviously the pepsin and the hydrochloric acid denature and degrade the proteins, and eventually you're going to get them down to amino acids. Well, it turns out that bacteria are able to actually convert some of their L-amino acids into D-amino acids, and they can actually incorporate those D-amino acids into proteins. Okay, a classic example of an area where you would find these is the cell wall, specifically peptidoglycan. Peptidoglycan is known for having D-amino acids in it, and obviously you only find peptidoglycan in bacteria for the most part. Okay. That's what we're concerning ourselves with, okay? So the bacteria that you ingest, have D-amino acids, and two very common D-amino acids are going to be D-alanine and D-glutamate, okay? And what we're going to do is we're going to look at a hypothetical here. What would happen, what would be the potential products if D-amino acid oxidase reacted with these amino acids, okay? And just bear in mind, you can't possibly avoid getting bacteria. It's impossible. It doesn't matter what you eat, what precautions you take, you're going to get bacteria. Okay, it's not like they're going to be super harmful, but you are going to degrade them down to D-amino acids. Okay, so let's deal with that now. Okay, so we have D-alanine here as the first example, and what I've done is I've essentially drawn you the initial product, and that's of course the Schiff base that D-amino acid oxidase generates. But keep in mind from the mechanism, that Schiff base, or we could say that protonated Schiff base, is going to leave the active site, and it's going to undergo spontaneous hydrolysis. And keep in mind the water in this case is going to attack this carbon. And my question to you is, what are the products? Well, if you're degrading D-alanine, well, you're going to get this product, right? If you have complete Schiff-based hydrolysis, you're going to get this and ammonia. And my question to you is, what is the first molecule that I drew? Well, that's just pyruvate, right? That's pyruvate, right? This is pyruvate. And we know that in some cells, specifically peripheral tissue cells, or we could say extrahepatic cells, they'll just run the pyruvate into the pyruvate dehydrogenase complex, and you can generate acetyl-CoA, and that will go into the TCA cycle. So that's one instance in which we can actually directly deal with the product, okay, and it actually benefits us energetically, okay. Now, one thing I do want to have you bear in mind with D-amino acid oxidase is that we are generating this very toxic product, which is hydrogen peroxide. Now, it's not like we don't have ways to deal with it. In fact, we actually have a way to catabolize hydrogen peroxide, and that's using the enzyme catalase. Okay, catalase effectively removes hydrogen peroxide. It's expressed at very high amounts in the peroxisome, and that's pretty handy because a place where D-amino acids tend to react with this enzyme is in the peroxisome. So nature has already given us a way to deal with that. Okay, now what I want you to bear in mind is that if you were to have just hydrogen peroxide going willy-nilly throughout the cell, keep in mind what would happen, okay? Assuming you're in the absence of iron, in which case you would have a Fenton reaction, hydrogen peroxide can just spontaneously do a homolytic bond cleavage. And what you would have after this reaction would be two hydroxyl radicals. And keep in mind that that hydroxyl radical on the oxygen, it's not tied up in any kind of resonance. There's nothing to shield it. It's very high energy, very reactive. And so if you just had hydrogen peroxide floating willy-nilly, you'd have spontaneous homolytic bond cleavages, and those hydroxyl radicals could oxidize proteins, nucleic acids, all sorts of lipids, things like that, things that are not good from the cell's perspective. So it's a good thing that we have catalase that can deal with hydrogen peroxide. Okay, but anyways, but yeah, so if we were to completely oxidize D-alanine, we're going to get pyruvate. And I don't think that's a big insight on the world. I think that you almost could have guessed that, right? We already saw what happened to L-alanine. That just reacted with L-alanine transaminase, right? And we got pyruvate. So I don't think it's a big surprise that D-alanine can get degraded to pyruvate, okay? We're also going to see something fairly similar in the next reaction. So in the next reaction, we're going to assume it has D-stereochemistry at the alpha carbon, and this is D-glutamate. This is D-glutamate. Well, I've gone ahead and drawn the Schiff base version of D-glutamate, and that is the product after D-amino acid oxidase does its reaction. Okay, and again, we're going to have Schiff base hydrolysis once this protonated Schiff base exits the active site of the enzyme, right? And again, we're going to have water that targets that carbon. And my question to you is, what are the products? Well, you should be able to predict Schiff-based hydrolysis products at this point, but if you can't, here's one product we get, and of course we also get ammonia. And my question to you is this: what is that first product that I drew? Well, you should be able to recognize that that's alpha-ketoglutarate from the TCA cycle. So assuming that we are at an extrahepatic tissue, generally what's going to happen is the alpha-ketoglutarate is just going to go into the TCA cycle. And also, even if we are a liver cell, it's just going to run into the TCA cycle, or it's going to react with various transaminases. Okay, so alpha-ketoglutarate, we can benefit energetically from that as well, but also bear in mind that we are generating hydrogen peroxide, but thank God we have a way to deal with that, and that's catalase. Okay, all right, the last one's not a D-amino acid at all, and my question to you is, first of all, what is that amino acid? And second, why doesn't it have D or L stereochemistry? And to answer the question, it's glycine, and glycine's achiral. So you can't assign a D or L just like you can't assign an R and S. Glycine is achiral. It has no stereocenter, so it's not D or L. However, it still can react with D-amino acid oxidase. Now, when you do the initial oxidation to the protonated Schiff base, you get this molecule right here. And my question to you is, what, again, are going to be the hydrolysis products when that molecule leaves the active site? Well, again, you're going to target that carbon. And when you do the Schiff base hydrolysis, again, you're going to get ammonia. I won't draw that again, but you're going to get this molecule right here. Okay, this molecule is an aldehyde. Okay, in fact, if you were to do D-amino acid oxidase on any of the D-amino acids, regardless what they are, this is the only one that's going to give you an aldehyde. And the reason it's the only one that's going to give you an aldehyde is it's the only one whose R group is a hydrogen. Okay, so all the other ones will be ketones. This is the only one where you'll get an aldehyde. And this molecule right here, this molecule is called glyoxylate. This is called glyoxylate. Now, glyoxylate really has one of two fates, and we'll look at those now. Okay, glyoxylate is an aldehyde. Okay, let's look at the first fate. Glyoxylate can actually react with a member of the aldehyde dehydrogenases, and recall that those are just simple NAD dependent oxidation. So let me do it like this. So initially you should have NAD comes in, gets reduced to NADH, right? And then water is going to come in, right, as the terminal nucleophile. And what you should get out of this is you should get something that looks like this. So we're still keeping this carboxylate right here, but this one should also be a carboxylate, right? And another way we could draw this molecule is like this. We could also write it right like this. Okay, and this molecule is called oxalate. This is called oxalate. Okay, and one of the things that's interesting about oxalate is, now whether or not this is a problem with D-amino acid oxidase is unknown, and quite frankly, it's an active area of research. But it's known that oxalate and its salts, which are calcium oxalate, so calcium can actually have an electrostatic interaction with oxalate. Calcium oxalate salts are a huge component of kidney stones. So kind of a question that you should sort of keep in the back of your mind is, could it be possible that something wrong with this pathway is part of the reason why people get kidney stones, or at least it's a contribution to kidney stones? Because we know for a fact that calcium oxalate is a major constituent of the actual solid mass of the kidney stone. Now, it's not to say that this definitely is the case, but it's certainly an active area of research. Okay, there's one other main reaction that glyoxylate can do, and glyoxylate can react with an enzyme called glyoxylate alanine aminotransferase. So this enzyme is called GLX, abbreviated GLX, alanine, and then transaminase. So transaminase. And just like all transaminases, this is a pyridoxal phosphate dependent reaction. Okay, now let's draw the structure of alanine. Let's draw the structure of alanine. So alanine is going to look like this. All right, so we're going to have our alpha amine, alpha carboxyl, and then the R group is just a methyl group, right? Well, remember what transaminases do. They're substitutions effectively, not mechanistically, but effectively between amines and carbonyls. So what happens if I go to glyoxylate and I replace this with an amine, right? Well, number one, I'm going to get this. I'm going to get this. What is this molecule? Well, that's just glycine, right? So you can say that one of the degradation products of glyoxylate catabolism through this enzyme is just glycine. What about the other one? Well, remember, they're effective substitutions between amines and carbonyls. Well, I have this amine, right? What happens if I replace that with a carbonyl? Well, I'm just going to get this molecule. And this molecule you should recognize as pyruvate. Okay, this is pyruvate. So ultimately, if I have a glyoxylate that I generate through D-amino acid oxidase reacting with glycine, you're going to get a glycine out of it. But effectively what this enzyme does is it takes alanine and converts it into pyruvate. Okay, so in the beginning of this scheme, we start with a glycine. We end up with a glycine, so there's no net change there. But it will effectively, through these two reactions, take an alanine and convert it into pyruvate. And we know that in extrahepatic tissues especially, that pyruvate can react with the pyruvate dehydrogenase complex, right? Pyruvate dehydrogenase, and then that will generate acetyl-CoA, which will go into the TCA cycle, and we can benefit energetically from that, okay? So these are the effective fates of our D-amino acids. They're going to react with D-amino acid oxidase, and then various other transformations can occur from there. Now, one other thing I do want to mention very briefly, and this is just sort of a takeaway message, okay? It was once thought, and maybe you still believe this yourself, and it might be possible that your teacher doesn't even know this, but it turns out that humans can actually biosynthesize D-amino acids. And there's only one that's known that we can synthesize, okay? And that is D-serine. So if you were to look at the structure of serine, okay, it looks like this. Keep in mind that serine's R group is just a hydroxymethyl group, okay? And D-serine, this is D-serine. D-serine plays an important role in the brain, okay? And I'm going to leave you there, and in the next video, we're actually going to look at the enzyme that makes D-serine, and it's called serine racemase. And in general, what racemases are is they're enzymes that reversibly convert between D and L amino acids, okay? And in fact, although it's not an amino acid, we've actually encountered one racemase before, and that was methylmalonyl-CoA epimerase. Okay, that's often named methylmalonyl-CoA racemase. Okay, and if you think about what racemase means, okay, racemase, what did you have when you had a racemic mixture? Well, when you had a racemic mixture, that meant you had a mixture of D and L isomers, 50-50%, right? So you had 50% D, 50% L. That's a racemic mixture, right? So that's where this enzyme gets its name from, that it's racemizing the isomers into the other isomer. In other words, it's taking D isomers and converting them into L, or the reverse reaction, it's taking L and converting them into D. And ordinarily, when we consider racemases, at least in humans, we're considering the process of going from L-serine to D-serine. And I'll leave you there. In the next video, we'll actually talk about racemases and their implications in humans. See you in the next video.